From Microbe TV, this is Office Hours for Wednesday, October 15th, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where you can ask a virologist, that would be me, questions about viruses or anything else that you're interested in, and maybe somebody in the chat can help us out as well. But there are no stupid questions. But there are stupid ideas, like, well, you know what they are. Welcome, everybody. Good to see you again. Hit the like button. Maybe we can attract some other people to learn about science here. Our moderators tonight are Les, Vanity Nutrition, Tom Steinberg, and I am sure you have noticed that YouTube has changed our handle names to the @ name. So here, Les Fabi was just Les Fabi before, this little part up here. But now it's his whole @ handle name, at Les Fabi 8997. Which, so supposedly it's for security, so people can't imitate each other. But for me, like, I want to call out people and say hello. What did I say? Hello, Les Fabi 8997. Well, here I know Les, so I can just say Les. But look, Brian Silverman 6695. So I know that's Brian, but he had another name. So I don't really like it. Tom Steinberg. I guess you could just make it @ Tom Steinberg, or without coast range either. Whatever you want. So you might have to change your name. So here, for example, Will has got Will Newcomb. So I recognize that, and that's fine. I could just say, Will is here from China 24C. Anyway, tell us where you're from. We will shout you out. Brian, by the way, is in Montreal. 8C and partly cloudy. Let's see what it is here. That's Roanoke. Because that's where I'm going tomorrow. Scotch Plains, 13C. So this is my fourth thing today. I had 2 podcasts today and 2 live streams. And after this, I'm looking forward to going to sleep. Not that I don't like doing it, but I'm tired. Anyway, on Tweevo this afternoon with Nels Eldy, my pick of the week was this opinion piece about Kennedy talking about circumcision and autism, putting his foot in his mouth like he usually does because he doesn't know what he's talking about. He lies. He misrepresents. If you want to read it, there's a gift link from Les. Thank you, Les, very much. Gallbladder is in Franklin, Tennessee, where it's 72F. Nice. That's very nice. Yes. Luna. Virus is Luna. Rima. Hello, Rima. That hasn't changed. From Iowa. You like from Iowa? Meow. Little cat thrown in there. Kang is from Chicago. 15C and cloudy. Now here's one I lament greatly, because Veronica was great. Privyet, by the way. So Veronica, why don't you just make it @ Veronica? You can change that very, very easily, okay? Unless you particularly like Jellicle Kitten, but I like Veronica very, very much, okay? 1C in Moscow. No sunshine until April. That sucks. You're all going to go crazy. Yeah, and see, Vanity Nutrition is here. Well, I missed Vanity, did I? She says, is Les Fabi 8997 a new YouTube handle? No, it's just that they made this stupid change. But see, Vanity had her handle @ Vanity Nutrition. So that's cool. Nothing changes for her. Here we have Anatomy Professor, Vancouver, BC. I think that's a good handle, right? Anatomy Prof. Very nice. EHS Talent. Previously, some of these were exactly the same, except EHS Talent didn't have the @ sign in front. And it was capital EHS, I think. Eastern Pennsylvania, 13C. Brian Varner, 3169. Brian, Knoxville, Tennessee, 20 Celsius. Very nice outside. Now here's another one that's totally changed. This used to be noir. Well, it still is noir, but you don't know it anymore. It's Artist Medical. Good evening, Artist Medical. Here at Tess Siegel, or just Tess, from Canton, Massachusetts, 48F. Why don't we test for poliovirus in U.S. wastewater? Okay. I can talk about that. Let me put it on the side. I'm going to mark it and come back to it and see who else is here first. I will get back to it. Dr. Leem is in Perth, Western Australia, where it's a sunny 14C. You see, HJF 3022, I mean, no. Doesn't cut it. So Tom writes, they didn't let me use my name the first time I tried. So there probably is some other Tom Steinberg. That's the reason. So you're not always going to get that. I mean, I've got Vincent Racaniello, because how many of those are there? I mean, there are a few. Biology Soon. I don't know if that was his original name. Phoenix, 78F, 25.5C. Yeah, why aren't people seeing me? I don't see my video here. Do people not see me? Yeah, okay. I can't see myself. Here, I don't know why. Let's just keep going. Where are we? Biology Soon. GSD Bio is here from San Marcos. That's Jonathan. He's in the autoclave dungeon, at least 37C. I'm going to restart my YouTube streaming app. Live streaming. Because I like to see what's going on. Connect streaming software to go live. What the hell? It is connected. An error occurred. So let's quit Chrome. Does this have something to do with the government shutdown? No, it probably doesn't, but I'd like to blame them. Sorry about this, folks. I do want to see myself because it tells me that the stream is working. Yeah, same crap. Same crap. Excellent connection. Live chat. Yeah, there we are. Lots of error occurred. But now I see myself. Good. GSD Bio. Jellicle Kitten has to move somewhere where it's plus 20C in the autumn. Many places for that. Now here's Mike. Oh, Mike from Puget Sound. Mike Dignacci. I don't know who that is. Puget Sound. I mean, that would be Mark Martin, but this is not Mark Martin. 58F on Puget Sound. Carol Wong, our favorite nephrologist, Bakersfield, California, 18C. Dr. Leem says after a successful trial on one patient last year with chronic drug-resistant Pseudomonas infection, my hospital is currently gearing up to treat other patients with bacteriophage therapy. That's marvelous. You going to publish that? Because we could do it on TWIM, This Week in Microbiology. Jan in the desert. Tucson 26C. Tractor Poodle in Bangkok. Whoa, 27 and cloudy. Should I come to Bangkok? Is it worth, I mean, is it worth me coming? Because I don't know that I would do a podcast there because I know there's a lot of virology there. Tractor Poodle. Brian Silverman read The Good Virus by Tom Ireland. It suggests that phage therapy may be an alternative to antibiotics in some cases. Does this make sense? Well, yeah, you just heard Dr. Leem, right? There's a long history of phage therapy, right? And it began in the early 1900s with the discovery of bacteriophage. The Eliava Institute was funded in Georgia, the country known as Georgia, and it has been working to this day. The discovery of antibiotics, around World War I put a little kibosh on phage therapy. But now with antibiotic resistance getting really out of control, people are re-examining phage therapy. And I think it does make sense, yes. I mean, there are some challenges for sure, and we've talked about them on TWIV. We've had a few episodes about phage therapy, notably episode 501 was in Texas A&M, College Station, where an amazing therapy has some history there. So, yeah, I think it makes sense. So, Veronica is very attached to Jellicle Kitten. Okay, you don't have to change it. I really am attached to Veronica. I like that name because Veronica, Veronica, right? That's fine. It's your handle. You can do what you want with it. Is it permanent? I don't know. I think people are going to scream because I don't think people like it. And we'll see what happens. It may not be permanent. Artemis. You see, here's Artemis 3263. That's Artemis. I know that in her name that she had before the at, it was ART. It was separated in some way. I don't remember exactly right. Hello from Montreal. I'm putting some stars on questions to come back to. Sandra Demet, hello. Welcome. Jeff, now this I recognize as just Jeff from Elgin, Illinois, 15C. Peter S. is here. Lagotic from Chicago, 14C. Pete Glass, connections issues in London. Don't think I can today. Sorry. Something odd. I'm sorry about that. Cheryl in Santa Rosa, California, 64F and cloudy. Peter S. is in Boulder, autumn day 18C. Mike and Lori Ding, thank you. Right. I see the ding in there, but Dignaci, is that the full name, the last name, which is very Italian? Okay, Mike and Lori. Nice. Nice to see you. Nice to have you back. I know Airy Ozone, 64F in Asheville. Come to Bangkok. All right. So some of you may know Amy, right, from Q&A with A and V. Well, she was in Bangkok a couple of weeks ago. Just for like 5 days for a WHO meeting. Jill is here from Wilmington, Delaware. YouTube is glitching, eh? Oh, so the handle is inspired by cats. There's a song about Jellicle cats. Okay. Michelle is in Philadelphia, 14C. Michelle, we didn't get together when I was in Philly for 3 days. Maybe you don't want to see me. That's fine. But I really liked Philly, and I want to go back periodically. Abdurrahman from Brooklyn, New York. Welcome. Mr. Doreen, 14C, outside of Chicago. MN is outside of DC. I got an MMR vaccine in January. Should I have gotten 2? Should I go and start another round consisting of 2? Well, it's a 2-shot series, right? It's a 2-shot series. So you should get the 2nd shot. Dean is in Wisconsin, where it's cold. Lori wants to speak with me regarding my eyes. She says, you got to speak with Daniel Griffin because he's the expert. I'm no doctor. I'm just a scientist. But Daniel would help. Questella, welcome from kind of Iowa-ish. YouTube has had outages worldwide for the last hour or so, and it's slowly coming back. People may see temporary weirdness until everything is fully back online. Don't panic. Give it time. Yeah, it's easy to panic, right? Especially when your program is on and you want to watch it. By the way, what do we have in store tonight? Some news, mini lecture, and chatting with you. Privyet, Elena from Reno, Nevada. Happy to have you. Yes. So myiasis is the solution to the last TWIP clinical problem. It will be released on Friday. It's kind of ooky. Yeah, go do your search and read about it. 11C in Reno, where Elena is there. So Redhead thinks the MMR for adults is once and done, but I think Daniel has always been talking about 2. Because if you haven't had any, you should have 2. Let's look. Yeah, it says, Cleveland Clinic says 2 doses for adults. Peter, I wanted to pass along that Cam Research says hi. Hello, Cam Research. Used to be here a lot. Jefferson Space from Miami. First dry air this week. Tractor Poodle says Jellicle cats come from a poem by T.S. Eliot. It's cool. Carol got the best birthday present, a healthy granddaughter. Congratulations. Very nice. And Mike and Lori ding. Yes, now I got it. Hopefully I can remember all this. I should be. Christina's in Houston and the largest cell and gene therapy manufacturing site in the USA. Mr. Doreen says, yes, you are a doctor, Dr. Racaniello, just not an MD. A doctor of philosophy. Most people, when they say doctor, I don't want to be a doctor, an MD imposter. I never wanted to be. So I don't ever call myself a doctor. Many people, like Mark Martin, this is Dr. Mark Martin. I would prefer professor because that tells you I'm a teacher. And that's what I like. Yes, but thank you, Mr. Doreen. The last name is Dignaci. There you go. Love it. Congratulations to Carol. Pete, had my flu jab today. I've dropped out of the age range for the COVID. Now over 75 only. I think anyone who wants COVID should be able to get it. It's not right because young people get long COVID, and vaccination mitigates against it. A 9-month time between the first and the second is okay. Let's see. Well, yeah, because in kids, second dose is given years after, so 9 months is okay. Although it may be soon, right? I don't think so. 9 months is enough for antibodies to go down. All right, let me go back to a couple of questions here that I didn't address. Here we go, Tom. Discussion point, difference between infectious dose and lethal dose. Inbred versus outbred host population, other variable factors. Okay, so infectious dose. How much virus is needed to infect the host? It says nothing about disease, just infection, right? You could, we do it in cell culture also. We do an infectious dose 50%, the amount of virus that will infect 50% of your cultures, your cell cultures, right? If you have 10 dishes with cells in them, the amount of virus that will infect half of them, and you can make dilutions and figure that out. But can also be in a host. You can do it in mice, for example. A lethal dose is just what it says, the amount of virus you need to kill a host. I don't think we apply that to cells in culture, right? Because although we do kill cells in culture, right, with certain doses of viruses, but we don't call it a lethal dose. We save that for animals. So there's a difference between the amount of virus you need to infect you and the amount of virus you need to kill you. And it very much depends, of course, on the virus and on the host that's being used. Since we can't really infect people for the most part, we have very, very little information on that. But in animals, you can challenge animals with different amounts. For example, you can give them a small amount of virus so that they will develop an infection and survive. They will be able to recover because their immune system is able to control the infection. But if you give them a high dose, the virus immediately is able to replicate extensively at many sites and may overwhelm the immune system. Of course, the thing to remember that animals are all different. Every human is different from another human, and the same is in the wild. Wild animals are all different from each other. Now, in the lab, we inbreed mice, right? We have inbred mice, which are genetically quite homogeneous. And we have different strains of inbred mice. And that does not approximate the variation that you see in natural populations, but it allows you to have a more consistent result when it comes to, say, infection, when it comes to immune responses. Whereas if you had outbred animals, every one is going to respond slightly differently. Very hard to study. But even with an inbred animal, if you infect 10, 20 mice, you're going to have different outcomes in different animals, even though they are inbred. And what controls the different outcomes? Differences in immune responses from innate all the way to adaptive and everything in between. Non-specific factors. What would be a non-specific factor in a mouse if it's healthy or not? It has other diseases which some mice can have. So, but in general, in mice, you get a nice homogeneous population, and you're hoping for as similar outcomes as you can, but there are always differences anyway. Okay, is that good? I hope so. Ask a question if it's not. I like to yammer. Okay, there was another one here. Here you go. Why don't we test for poliovirus in the U.S. wastewater? So let's go back to 2022. So the U.S. has not had a case of poliomyelitis, the disease, okay, since 1979. And suddenly there's a case in Rockland County, New York, and a 30-something male who develops poliomyelitis. And this male was part of a Hasidic community or an Orthodox community and was not vaccinated. So they get virus from him, and they show that it's a type 2 vaccine-derived poliovirus, which came from somewhere else because we don't use attenuated poliovirus strains in the U.S. anymore. The Sabin strains, we don't use them. So someone came in shedding that strain, someone who had been vaccinated elsewhere or had picked it up elsewhere, and it got into the human population in the U.S., and then the CDC went and looked in the wastewater in the New York area and indeed found it in many locales. And we had a mini lecture a while ago about this this idea. So they only did limited wastewater sampling, which showed that there was circulation in the New York area, which is not surprising because virus comes from overseas into New York, which is a busy port, right? Get many, many different countries coming, people from different countries coming into New York. So then the question is, what about the rest of the U.S.? And the CDC doesn't let you do that. You can't sample your wastewater for poliovirus. They say it's a biohazard, which is complete and total BS. It's not a biohazard. Because even if, why would it be a biohazard if you're looking for poliovirus as opposed to not looking for it? Let's say you're looking at wastewater for SARS-CoV-2. If it's got polio in it and it were a biohazard, it would be a biohazard then, not just when you look for polio. So that's BS. I can't stand when people lie like this. And then they said it wouldn't be informative. Of course it would be informative. More BS. You would need to know where the virus is to make sure that people are keeping their immunization rates up. Because when they dropped, like they did in Rockland County in 2022, you get poliomyelitis cases. And so if it's circulating in Chicago, we need to know this. But we don't. They won't let you do it. And if they're doing it themselves, they're not telling you. And I knew some people in state health departments that were doing wastewater sampling and wanted to include polio, and the CDC told them, no, you're not allowed to do it. Now, of course, who knows what authority the CDC has anymore? They're all broken, right? So I don't know. I don't know what to do. I don't know what authority they had to begin with, right? They don't have any legal authority. They can only make recommendations. So in my opinion, the problem is the state health departments don't want to go against CDC because they give them some money, right? They have some money coming from the federal government via CDC. So they don't want to get them upset, right? Because they'll turn off the money. And now in this administration, you get them upset and they'll just do all kinds of things like suing you for an $18,000 mortgage payment or something like that, which has nothing to do with reality. Okay, so that's why we don't test, because the CDC says we shouldn't. When I say many other countries test for poliovirus, and we need to know what's here, because that tells us the extent to which attenuated viruses, polioviruses, are coming from other countries and are circulating here in the U.S. Wouldn't you like to know? I would. I'm a virologist who studied polio all his career, so I want to know where it is, and you should too. So that's the answer. And I think there was another question that, yeah, here we go. Kang says, how do we determine if there are phages for Legionella without a PFU assay? Okay, so this is the paper we did on TWIV last week. And they tried to plaque this new Legionella phage, and it didn't work. So what they did simply was they added virus to cells, to bacteria, Legionella cells growing in culture. And they saw, for example, that the phage would stick to the bacteria and that it would reduce the number of bacteria in the culture. So it was killing the bacteria Actually, it's a temperate phage, so I'm not sure it was doing it, but it was sticking to it. Kang, I'm going to go look at the paper right now because I have it here. Let's take a look and get this because my memory is not great on that. Microbe TV, TWIV, that would have been TWIV 1261. Yeah, there we go. The discovery of a Legionella phage. So, okay, how did they do that here? They couldn't do plaque assay. Where is it? So we asked whether these particles are infectious. Traditional plaque assays did not generate any plaques. So they developed two assays to test susceptibility. An attachment assay and an infection assay that measures lysogen formation. Okay, so in the attachment assay, you add virus to the Legionella, you let it absorb, you wash away, and you ask, can you detect phage sticking to the surface of the bacteria? So they found that the phage was sticking, so that's good. And then they looked at the effect of the phage on Legionella replication. So they measure growth, and if they assess the impact of the phage on the ability of Legionella to grow inside human macrophages, and exposure to the phage reduced the intracellular growth of Legionella by up to 20-fold. Okay, so those are the two things that they established. The other thing is the lysogen assay. So you infect with the phage, and then you look in the genome of the bacterium, of Legionella, because the phage, being a temperate phage, doesn't lyse the cell, but it integrates into the genome. And so you can measure the integration of the DNA in the genome. You take DNA from the bacterium after infection, and you measure integration of the Legionella phage. So that's how you do it without a plaque assay. It was a very clever paper. Yeah, very good. Okay. Janity Nutrition says, I just saw California made it illegal to call yourself doctor unless you're a physician in a healthcare setting. Yes, I don't call myself doctor. Never. I'm professor. But in Germany, what they call you, what I would be called in Germany would be Herr Dr. Professor Racaniello. And I can show you because I bought a train ticket once in Germany and it said on it, Herr Dr. Professor Racaniello. Because they have respect for their professors over there. Oh, gosh. Okay. Let's see. Any other questions? Yeah, because I want to do some news also. So, Eric says, I had the idea that one MMR is okay for folks who got the original measles. If that's the case, you could get just one, but I don't know if that person had measles, right? Not sure. MN 9159. I reached out to you via your email. I'm sorry. I will go search and answer you. I'll give you Daniel's email, and you can reach out to him because he's the doctor. He's the medical doctor. Artist Medical wants to know, is the chance of getting chikungunya from a mosquito in Nassau County a thing now? So a locally acquired case of chikungunya was confirmed in Nassau County on Long Island. And so this lady had not traveled anywhere, and she developed chick, and she tested positive for chick, so she must have gotten it locally. Is it a thing? I don't think so, because, the mosquito season is ending here in the Northeast. It gets cold, they go away. And just one is not an issue. So I think we'll have to wait till the spring. So right now it's not a thing, no. No, that's a good question, yeah. So gallbladder 8 is an MD. Says chiropractors, optometrists, and some PhD holders have no problem using doctor. And many, if not most patients, are unaware of the differences in training. Yeah. As I said, I don't use it. I'd rather be a professor. Or just Vincent. You can call me Vincent. It's no problem. I am not defined by my degree. Yes, a chick has been detected in the U.S., Long Island, one case. Up to code Danny, delusions. Reminds me of a 1981 memory titrating in first year chemistry class. I could never get the hang of it. The solution in the beaker would usually turn bright pink. Yeah, I love titrating. For viruses, we have to do that all the time. Ariozone wants to know if the current community spread of conjunctivitis is a sign of adenovirus or if there's a specific virus solely causing pink eye. So it could be adenovirus, it could be an enterovirus, and some other viruses as well. Adenovirus, varicella zoster, herpes simplex, measles, and of course rubella, Epstein-Barr virus, and even a bacteria, chlamydia. So it could be many things, Ariozone. Okay, call me Ishmael for now. Cool, 16C in Maryland. Do you have any idea if MMRV is effective against shingles? No, I don't know, because it's given to kids to prevent chickenpox, right? The varicella component. So we wouldn't know about shingles, no. Yes. Just don't swim in the wastewater like RFK, talking about polio in the wastewater. CDC is now short for the counter with donuts and cake. With respect to polio, a friend of mine had polio as a child and didn't suffer paralysis, but as an older adult now suffers partial paralysis in one of her arms. That's very interesting. So they had a non-paralytic case, which happens 95% of the time, 99% of the time, yet they still lost some neurons apparently, and now as they're aging, they lose more and you get post-polio. Interesting. Yes, always better to be vaccinated, for sure. We have 119 people here and only 32 likes. Please hit the like button, folks. Please hit the like button. Carol enjoyed Immune Booster with Liz Marnick. It was very good. I like that Cindy wants to go and do these interviews at meetings and also in her office at Cornell. It's great. Why are a bunch of CDC people getting fired during the shutdown? Well, here, let's do the news. Some news, okay? Here, let's take Melinda off here. And the first news story I had for you today is from, actually, there are many, but I picked one in Time magazine because it's pretty cool. Because my former student, Angie Rasmussen, is quoted extensively. CDC not functional after Trump administration orders mass firing. So Friday, many hundreds of people were fired. 1,300 employees received notices, right? 70 epidemic intelligence officers, the entire staff of the publication Morbidity and Mortality Weekly Report, and others as well. Then they realized that they made a mistake, and hundreds were said, "We didn't mean to fire you. You have your job back." Now, the reason they were fired in the first place is because, as you know, the U.S. government is shut down. It's not funded. And so normally you would furlough these people. You'd put them on leave and they don't get paid. But Trump is taking the opportunity to fire them all, which is something he does not have the power to do. It's being challenged in the courts. It will probably make its way to the Supreme Court, and they, in their backwardsness, will probably give him the power to fire any federal employee at his whim. So this is really, really screwed up. But the effect on CDC, of course, is they have problems detecting outbreaks and figuring out what's going on, not just in the U.S., but all around the world. So this is horrible because of this effect, but also shows how this administration doesn't do any work at all. They just fire people without looking to see if they're the right one. They said their positions weren't coded properly. Well, you know what? It's your job to find out that they were not coded properly before you push the switch. You're just a moron. You're a frickin' moron. And so was Musk when he just randomly fired people just to fire them and not realize who is important. They're all very important, but some shouldn't go. Like MMWR, Morbidity and Mortality Weekly Report. You shouldn't get rid of that. It's a fantastic outbreak reporting journal. And I was very upset when I heard that, so I'm glad it's back. But the other thing is, besides all this, when you fire a person, it's a traumatic event, right? Here they are working, and then you say you're fired because we don't need you, and that really depresses many people, right? It really takes a hit on their confidence. And you shouldn't be messing with people, especially if you're going to turn around the next day and say, sorry, it was a mistake. But they actually didn't apologize. They're shitheads. And finally, they announced this on a Friday, so it's not going to get covered by the news for a while. They're just freaking cowards, every last one of them. And it's disgusting to see them having their way with really good institutions in this country. Now, another one here. This is the hypocrisy of this administration. Here's a story in NBC News. Trump gets a COVID vaccine during his checkup. Well, how about that? How about that? He got his COVID vaccine. His doctor said he got his COVID vaccine. All right, so why am I on about this? Because he's the one who hired RFK Jr., who says no vaccine works. And in particular, he says the COVID vaccine has killed more people than it saved. And yet the president of the United States is getting the COVID vaccine. So what is it? Is it good or not? Is it safe or not? Well, of course, Trump's doctors realize he's 79 years old. He should be protected. And the vaccine is the best way to protect him. And that says that RFK Jr. is full of it. He doesn't know what he's talking about. He's lying. Yet Trump supports him. He's still there. And he's silently agreeing with what he says by not dissenting. Oh, my God. What a hellhole we are in these days. All right. I think I have one more news item here. Oh, this is another one. Yeah, I have two more. Measles cases continue to climb. This is an NPR story by Maria Godoy. With outbreaks across the country. 1,596 cases this year, the highest number in more than 30 years. And Paul Offit says it's probably closer to 5,000 because there are a lot of cases that are not properly diagnosed. Okay. And there's an outbreak in South Carolina where 139 school children in 2 schools now are quarantined for 21 days. Transmission in Arizona, 77 cases so far. In Minnesota, there are cases. So soon we're going to lose our measles-free moniker. We're not measles-free anymore, right? It was declared eliminated in 2000, and now we're back because RFK Jr. and his ilk will not push vaccination to stop this outbreak. It could all be stopped, right? All you have to do is send vaccination teams to these areas, vaccinate people. That'd be the end of it. But he has this ideological belief that vaccines don't work, and he's a moron as a consequence. Okay, one more thing. Immune 100 live at the incubator. So on December 22nd, we will record Immune 100. We'll have Cindy and Brianne and Steph and myself. We will have 2 guests. So this is the Eventbrite listing. You can get to this by going to the microbe.tv webpage. There's a link to get here. December 22nd, 6 p.m. Doors open at 5:30. It's going to be live, and it's going to be probably live streamed, but you have to buy tickets, right? So if you go here to get tickets, there are different tiers. So we can only accommodate 25 people. So probably local people are going to want to come. Maybe we have a couple of $25 levels for students. Then we have $50 and $100 and $250 and $500, limited numbers of each. But you get different amounts of swag. You get admission, you get refreshments, you get stickers, you get mugs, you get postcards, you get posters, you get T-shirts. You get all these things. But you get to hear the presentation. So that should be a lot of fun. And if you're in New York and you can do this, we'd love to see you. But you should sign up sooner rather than later because it's only 25, right? Only 25 seats available in that little incubator. Okay. All right, let's go back to here. So we had just answered Melinda. Why are a bunch of people getting fired? Because of the government shutdown. Will says, Herr Dr. Professor Racaniello, respect. They respect professors in Europe. There's just no two ways about it. Here we do not respect professors unless you're taking their class. Then you have to, so that you get a good grade. Jill said, my old co-worker's last name is Dreyer. In Germany, everyone called him Herr Dreyer, which we all found amusing. That's very funny. I like that. MM is not sure of either measles or an early vaccine. So in that case, get 2 shots. That's all there is to it. Not going to harm you at all. In Germany and Austria, doctor would be part of your official name on your passport. My passport just says Vincent Racaniello. That's fine. Tractor Poodle in Thailand, they will call you Dr. Vincent or Professor Vincent. That's fine. Professor Vincent is okay, or just Vincent. If you want to have a respectful, handle, Professor Vincent's fine. That's okay. Now, Kang says, I talked with a friend who caught COVID a week after being vaccinated for the virus. Her doctor said she was weakened by the vaccination. No, wasn't weakened by the vaccination. A week is not enough to get an immune response, okay? That's all there is to it. Is there enough interferon produced to block an infection? By vaccination? No. The infection itself should generate enough interferon to mollify it, but everybody is a different interferon responder, so she may not have responded well with interferon. But the point is, you need about 2 weeks after the vaccination to get optimal protection against disease, and it wasn't long enough. That's all there is to it. Okay? Yes, weakened by vaccination is nonsense. It's absurd. You don't get weakened by vaccination. What does that mean? Tractor Poodle, you can host a podcast at Chulalongkorn University. I would love to find some local virologists and have a chat with them. That would be great. Can you hook me up, Vincent at microbe.tv, with some people who might be interested? Yeah, Angela has her own outstanding Substack. She writes very well, and you should check it out. Angela Rasmussen Substack. Yep. The GOP wants to show they can run things without the government. Oh, yeah, it's running really well. try and take a flight. I have to take a flight tomorrow. I hope it's okay. Yeah, we want America healthy, so we fire the CDC. It makes perfect sense. Well, they don't want government level institutions anymore. They don't like that kind of control. I saw a wonderful, this has nothing to do with this, but I saw a wonderful Instagram by a doctor, and she played a little clip of J.D. Vance where he said, do you ever go in an emergency room and have all these foreigners in there, and why are we giving them health care? And this doctor lady said, I got news for you. We went into medicine to help people, and we help anyone who's ill. Doesn't matter where they're from, and you're never going to stop us from doing that. I thought that was brilliant. I love it. I mean, what is this elitist crap attitude that you can't take care of people from other countries? I don't care where they're from. If they're sick, take care of them. Doctors will do that. Okay? It's not taking health care away from anyone else. Oh my God. Yes, Mike says to Lori, I've done telehealth appointments with Dr. Griffin. I will send Dr. Griffin your email, and he will get in touch with you, and you can do a telehealth, okay? Lori. Jeff writes, America's conservatives have wanted to destroy the U.S. government for decades to push functions to the states. This stems from their pro-slavery days when the admission of free states was a threat. Tona, you're back? I thought you were away. I liked Professor R's rapport with the Belgian scientists on poop from 32 planes. Yes, I know Elke for a few years, and it just was in a casual setting, and I've talked to her a lot, so we had a nice rapport. Yeah, and I met her years ago, and I love this story, and I said, when it's published, we'll get you on TWIV, and it hasn't been published yet, so I said, let's talk about it. So that was good. I had fun. Yeah. Artemis, I heard someone say that antibody-dependent enhancement may make you more prone to the disease. No, well, it depends. 2 weeks, there you have a good immune response already. It could happen with, say, dengue, but we're not vaccinating against dengue here in the U.S. But if you got immunized with a dengue vaccine, well, if you had all 4 serotypes and you got a good immune response to all 4 serotypes, it shouldn't be a problem. Okay, so in theory that could be a problem, but it isn't. Now, the problem is in some other countries where they were testing Dengvaxia gave a poor response to the type 2, and so they could get antibody-dependent enhancement of type 2 on a subsequent infection. Hello, Margot in Phoenix Metro, 75F. Spamlet, enjoyed your talk of human microbiomes recently. Yeah, we talked about it on TWIM, I guess, where it's linked to frailty and poor sleep quality and frailty. Yeah, I'm interested because I don't sleep well. And so I wanted to know this study. Google's AI told me that our gut has bacteria with cellulose digesting ability. That might give us some ruminant-like energy. Well, we're not ruminants because we don't have more than one stomach, right? But some of the bacteria in our gut certainly help to digest plant material. And did you know that if you live in the U.S. and you eat seaweed, you're not really good at digesting it. But if you eat enough, the seaweed has bacteria on it that will colonize you and enable you to digest it. I saw that talk years ago at a conference. Getting rid of jobs helps the economy. Oh, yeah. Hello, Visto. Welcome. Frickin' moron. Did I say that? He's a frickin' moron. They're all frickin' morons. None of them. I don't like to be ad hominem, right? That's a personal comment, a criticism, because you have to focus on what's the problem, right? But they get away with so much because they're lying and they don't know what they're doing. So, yeah, that's it. They're frickin' morons. Just in a general way. I'm not personally insulting anyone. Thank you, Jason. Yes, No Kings, October 18th, 2025. Yep. Vaccines for me, but not for thee. That's the thing. Yes. Very good, MN. Fired people won't return after that treatment. Yes, they shouldn't, because it's insulting to them. Get another job, yeah. Hello, Crystal from Edmonton, Alberta, where there's a lot of measles also, right? Jeff, the Republicans want to make federal employees miserable. They've said this directly. Search for vote and traumatizing federal workers. They are no good Republicans. And you know, the House and the Senate are all colluding to not pass the appropriations bill. You know, the Democrats want restoration of Medicaid cuts, and they want discounted Obamacare, and the Republicans will not give them that. They will not give that to the American people who deserve it. It's disgusting. Why do you want to take away health care from people, right? People deserve health care. That's what a government is for. In the UK, we say a load of bollocks. Yes. How about a positive story? A phase 2 trial, an allergy spray showed promise. Yeah, I know which one you're talking about. We covered this on Daniel's clinical update, and really the effects are marginal, and Daniel said he would not prescribe it based on those results. Visto wants to know, when measles virions are spread all over a building, how long do they last? That's a good question. Let's look it up. It says 2 hours. 2 hours. Not a long time, but doesn't take long, right? Yeah, ezolastine spray, right? Kind of moderate effects. I don't know what this refers to, Jason, but it's a good saying. Exploring and tinkering with the beautiful programming of life. I like that. Try not to think about it. Your quote on the recent clinical update about when you put a clown in the palace was just the best. Yeah, thank you. I don't remember it, but thank you. I'm glad you liked it. Vanity Nutrition, who's a flight attendant, says flights should be fine. So I hope so, Vanity. Tomorrow. Jason says that Nano Room's new series, The Essence of Biochemistry, gorgeous, smart, talented science visualization and exploration. Sounds great. I will look it up for sure. Margot says Saudi Arabia took care of my mom when she had an unexpected seizure on a boat. She was at 2 different hospitals, 2 MRIs, glioblastoma, found surgery in the States, though, but they took care of her. Yep. Yep. It is my understanding that Medicaid reimburses hospitals for patients who are treated for free, both citizens and non. Yeah, it's a low fraction of the cost, I guess. But I don't believe that you can turn away anyone who comes for help, right? The doctors out there, please confirm or deny that you're not allowed to turn away a patient seeking care. And that's what this MD said on Instagram. But on Instagram, anybody could be a doctor, I guess. Tractor Poodle, what's the status of Columbia University's collaborations in Ukraine for antimicrobial resistance? I don't know. I didn't know that anything was going on there. Let's look it up. So, Columbia University, through its International Center for AIDS Care and Treatment, is providing technical assistance to Ukraine to combat antimicrobial resistance. It supports development of plans, strengthening infection prevention and control measures like hand hygiene, and building Ukraine's laboratory capacity to monitor and respond to antimicrobial resistance, especially in the context of the war. So that's what's happening. Yeah, I didn't know about that. Vincent, magnesium supplement helps with sleep. I take magnesium glycinate at night. Yes. So Saturday I started taking magnesium glycinate. I don't. I have a sense that I'm sleeping better, but I still wake up at 4 a.m. no matter what time I go to sleep. I can usually sleep, let's say, between 11 and 4. But then at 4 I wake up and that's the end of everything. I can't get back to sleep again. I think the magnesium has helped me get a solid 11 to 4, whereas before it was not so solid. But we'll see. We'll give it some time. I do feel a little bit better. So, yeah. Thank you. Keith is from Long Point First Nation in Winnoway, Quebec. Thank you for including me in your time zone. Here, Dr. Professor Racaniello, I've been invited to teach high school science up here. Lovely. Great to hear it. Nevitz wants to know if the number of microbacterial cells outnumber the number of human cells in an adult body. Yes. Not as much as we used to think. We used to think it was 10 times, but it's not that much anymore. I forgot what the number is. Yeah, I forgot what the number is. Aubrey says, sometimes dumbasses are dumbasses, but usually not leading the federal government. It's true. It's true. Oh, yes, Kang, could Dan Wilson and Dave Farina be invited to talk about their debate against the anti-vaxxers? I don't know. Did anyone watch? Did anyone know how it went? Let me know. Eric met a MD PhD who was a Frau doctor in Germany. That's cool. Yes. Love it. So Jill says they did a heroic job against Corey and Kirsch. That's great. Spamlet says, have you or Diana thought that it might be possible to set up an online clearinghouse for difficult to diagnose cases? Could be rewards from national services and insurance cost for solving. I haven't. No. It's not my purview, right? It's Daniel if he wants to do that. But if you want to run it by him, send him an email at daniel@microbe.tv and propose it. He may not have thought about it, right? Concerning the nasal COVID vaccine, what about the T-cell response? We know that is critical. Let's look it up. I don't remember. Yeah. So they didn't look at T cells yet. They're probably going to, but as far as I know, in the one report, they just looked at antibodies and they seem to make a good antibody response. But, you know, it's not going to be sterilizing because memory takes a few days to kick in, all right? So it's not going to sterilize. I'm sorry. You can dream all you want. It's not going to sterilize. The UK is using single-dose Gardasil 9. Let's see. Yeah, they dose. The number of doses depends on the age, right? Younger kids get 2 and older get 3. So 1 is unusual. I don't know what the data are for that. Gabriel, hey, all 17C in Greenville. I just finished the second molecular biology exam of my PhD today. The classes are interesting, but I wish I could get in the lab more. Good. You'll get in. You got to get those classes done. It gives you some good foundations. Yeah, when people can't get health care, they go to the ER to get free health care, which is even more expensive, and then hospitals get shut down. I understand that. But, yeah, I mean, they should be able to, but many people cannot get primary health care, right? And that's the problem. Polka Dot Puck got my flu shot, $10 off, bought 40 daffodils. Now I'm protected, and my ground is too from voles and gophers. Lovely. Lovely. Speaking of measles, what's this immune amnesia thing? Well, measles virus reproduces in lymphocytes, and that includes memory B cells, the cells that make antibodies and that stick around after an infection, and they remember the infection. So when you get infected again, they can quickly make antibodies. So in 2 days or so, they can really damp down the amount of virus. So measles virus, by killing those cells, wipes out all of your immune memory to all the vaccines you've ever had and also to any infections you have had. So you recover from measles and now you're not immune to anything anymore. You have no memory. You have to start over. That's really bad. And that's why when we had a lot of measles in the U.S., 500,000 cases a year, a lot of kids died of secondary bacterial infections because the measles infection was wiping out their ability to make antibodies against the bacteria. Yeah. So Costello says most doctors refuse Medicaid. And she says, I've never had a doctor in my life because of it. Well, Costello, you know, you are young, and when you get older, you're going to have to see a doctor because things break, I have to tell you. Visto says in Australia, illegal migrants get medication, vaccines patched up by docs, and maybe after sent to a prison island. Okay, that doesn't sound too good. And Jeff writes, Costello, believe it or not, Nixon suggested Medicare for all. That's the answer to our healthcare problems. Nixon, a Republican. How about that? Okay, Lori writes, I'm an RN for 30 years. You may not turn anyone away from the ER for treatment. All right, I'd like to know what J.D. Vance is going to do, right? And yes, Medicaid funding is getting cut. And this is what the Democrats are trying to restore to the funding bill. And the Republicans will not negotiate on this because they just want to take it away. Why? Because they're told to take it away, not that they really want to. Many people are saying the ER cannot turn away patients. There's another one, and here's another one. You cannot deny help. Good. Yes, emergency rooms are very expensive primary care facilities. They should not be used at that. But in New York City especially, they are used often for that, yeah. I do really well with 5 grams of melatonin. I don't have a problem falling asleep. It's just staying asleep. And melatonin, I understand, is good for falling asleep, but not maintaining you sleeping. But thank you for the suggestions. You came in late. They're using YouTube switched from using your names to using your actual handles, the @ name you got when you first signed up. So Visto Tutti would have been the name here before, but now it's the whole thing. They switched to it. Who knows why? I don't know why. Toner with the pancreatic cancer oral microbiome study. Did they keep taking oral samples through the years or a one-time initial? Oh, that's a good question. Let's look it up. Did they find other health morbidities? Well, they didn't report them. But let me look at the paper. I hope I have it. It should be in the TWIM. I keep all the papers that we discuss on all the podcasts. It can add up. It's just Dropbox is a pain because things are not local. Make available offline. Sleep quality, that's not it. Oral bacterial and fungal microbiome. Okay. I have to open this up to find out. It's a good question. I want to answer it. Now I have to make it bigger because I can't see it. Okay. Oral wash samples were collected from 2001 to 2002. I don't think they had more than one. I mean, they had 52,000 participants. So that's a lot of wash samples, right? They had to analyze each one. They provided oral samples. I think it was just one. And they didn't talk about anything else. No other risks. That wasn't part of the study. Okay. Now, this is a joke, right? I'm on ivermectin now and never have to take Paxlovid again. Now, that's not right? Jeff says the U.S. spends about $12,500 per person, while Norway spends $8,700. We have a lower life expectancy. Our system is low value. Yeah, we do spend a lot, and we have a poor life expectancy. I don't know why. I have insomnia from watching too much microbe TV. Maybe I have insomnia from producing so much microbe TV. No, I don't wake up and worry about microbe TV. I worry about other things. I mean, that's the problem. I worry about too much when I wake up. I can't get back to sleep. Just dropping in to donate via the link on screen tonight. Thank you, John, for your support of science communication. Likes. Come on, folks. We're way behind in the likes. Please. I'm not asking for any money. Just hit the like button. We have 141 people and 71 likes. It's a 50% return on investment. Please hit the like button. So Tona says the debate turned into a shit show. So I don't know. Some people said they did great, but I don't know. Mike, Obamacare provided for Medicaid expansion, but many of the red states refused the expansion. Yeah, it shouldn't be a political thing, right? Healthcare, no. China, primary healthcare comes from outpatients departments in hospitals. Yeah, I've seen some videos about people who go to a hospital in China for their care. You know, you make an appointment and you go in and so forth. It's part of the system. That's good. The low-dose naltrexone will help more with the going back to sleep and improve the quality. Yeah, it's a prescription med. Peter, I've had poor experience with melatonin for maintaining sleep. It can also cause a more assertive stature when dealing with others. I can't take it anymore. I'm going to give the magnesium a try. We'll see. In 1986, Vanity writes, Reagan signed the EMTALA, ensuring anyone going to an emergency department will receive screening and be stabilized for an emergency medical condition regardless of ability to pay. Yeah. EMTALA. Yes, debunk the funk debate. That's what we were talking about before. It turned into personal attacks. That's not good. Well, so tonight's theme is healthcare. Blue screens aren't great for sleep. Yes, I know. So here I'm going to be blue screening until 10 p.m. It's 9:17 right now. And you need a couple of hours of no blue screen, right? So I'm automatically screwed. But I do fall asleep. That's the thing. It's not falling asleep that's the problem. It's just that I wake up. Will is on magnesium glycinate plus a handful of walnuts an hour before bed. So Biology Soon says YouTube switch to unique handles to reduce impersonation, make channels easier to find and tag, and enhance security. Okay. It's not as much fun. Peter asks, life expectancy is hugely tied to diet and physical activity. Americans overall tend to eat poorly and not exercise. Yeah, but what about the difference between men and women, males and females, which we talked about on Tweevo today? You know, it's more than just that. I mean, that may be part of it. But in birds, the males outlive the females, whereas in many mammals, the females outlive the males. But not always. Not always. I know someone who had acute lymphoblastic leukemia. He was successfully treated with immunotherapy, but had to have all his vaccines again. Yeah, because they're B cell. To kill all the B cells, right? A neurologist or psychiatrist can help with sleep issues. Also, maybe you have sleep apnea. I do. I've been looked at. I have sleep apnea, mild sleep apnea. But I'm not wearing a frickin' thing. I'm sorry. Or just sleeps like a rock. Yes to the magnesium. Oh, meditative videos will calm the mind. I heard brown noise is also something good. Right? Brown noise. Where's the like button? If you're on a phone, someone with a phone, tell Lori where the like button is, please. So I appreciate all these suggestions. Gabriel, maybe white noise in a cool room, no screen for 2 hours, and viewing sunlight as it rises and falls. Oh, this would be perfect. Yeah. All right. In Thailand, we have private clinics, but most people get their primary care from OPD clinics and public hospitals. Siri Raj Hospital, Bangkok, treats about 10,000 patients a day. Wow. I don't know. That seems like a good thing instead of going to private docs, right? Brandy works well for sleeping. Yeah, right. It's gonna do other bad things to you, though. Hello, Lise. Welcome. I hate it when my brain blue screens. Yes, the blue screen of death. Meditation. Yeah, I think so. Our former moderator, Steph, suggested that, and I liked it. I tried it for a while, but I stopped. It's just too much noise around. Maybe that's all the sleep you need. Well, like right now, I'm not tired at all because I'm jacked listening to you. But if I'm in a quiet situation and I'm reading, I will fall asleep very readily. Or if I just sit in a chair and start thinking, I can fall asleep immediately. So I obviously need some more sleep than I'm getting anyway. Let's see. If you're on the phone, scroll the chat down so you can look under the title, under the video. You'll see a like and dislike to the left. There you go. Will wants to know, males have more COVID fatality, like twice as much, but females more long COVID. I don't know why. We don't know why, but I could speculate that, the females somehow have developed mechanisms to avoid fatality, but in so doing, they convert it to a persistent symptom issue, right? That's what I'm thinking, but I don't know exactly the mechanisms. No. Gabriel says, I research HTLV and want to develop a better model. HTLV lies latent for many years, so models don't represent its long-term effects. So we just did a paper on TWIV 2 weeks ago on a group that developed a humanized mouse model for HTLV, which mimics latency. So go check that out, Gabriel. Yeah. Pete says, if I close my eyes trying to concentrate on Vincent's mini lecture, I tend to nod off. It's 2 a.m., of course. Ay, ay, ay. Melatonin tends to induce insane dreams. I don't need that. And hydroxyzine, you will sleep like a baby. I don't know what hydroxyzine is. I'm glad for you that sleep well. It's wonderful. Now, speaking of sleeping, I'm going to put you to sleep with a mini lecture. Okay? We're going to continue the rest of synthesis of RNA from last time. All right? We were talking about plus-strand RNA viruses, and we had finished with poliovirus, a plus-strand virus. The whole point of this mini lecture is not to put you to sleep, but to have you understand a bit how RNA viruses make more RNA, how they copy their genome. So for Flavi and Picornas, it's just a long plus-strand RNA that's copied to a minus and then back to a plus again. Now for the alpha viruses, same idea, except there's a shorter mRNA made from the negative strand. Not clear to me why it works, so it survives evolutionarily. All right. So let's look at how this works. These are the Togaviridae. The viral genome is mRNA. For both of these viruses, the genome is mRNA, but this part of the genome at the right end is not translated. And that's where the mRNA, the subgenomic mRNA, comes into play. So that's what I'm calling it, a subgenomic, which means it's not the whole genome. It's a sub part of it, right? So these viruses infect cells. They attach to receptors. They get taken up by endocytosis. The RNA is released into the cytoplasm, and it's plus-stranded, so it can be immediately engaged by ribosomes, and they make a long protein which is made as a polyprotein, and it's cleaved by proteases to form the RNA polymerase. The RNA polymerase then copies the incoming plus strand to a minus strand, which then gets copied back to a plus strand. And the messenger RNA, the subgenomic messenger RNA, which is made from the minus strand, encodes the structural proteins. So when the virus RNA first gets in, you don't make any structural proteins, you just make the polymerase. And then later, when you have lots of new RNAs made, you make the structural proteins, and those can go on to build new virus particles. So if you want to know the functional reason for only translating part of the genome initially, you could say there's no point in making capsid proteins until you have genomes to package in them. And so, well, that's a human interpretation. Who knows why, what were the evolutionary pressures that led to that? So this is another view of what I just told you. Here's the plus strand RNA. When it gets into cell, the left-hand half is translated to make the RNA polymerase. There's a stop coding, codon, about halfway down the genome that prevents the rest from being translated. And so once the polymerase makes a minus strand, then the polymerase can make a subgenomic mRNA from the minus strand, which gives rise to the structural proteins. It's a little bit Baroque and obscure, and it just is an illustration of how you can have different patterns, and it's not clear the reason, the functional reason for them. Now coronaviruses, of course, are also plus-strand RNA viruses. They have a very unusual method for making messenger RNAs. So the coronavirus genome is a very long RNA, a plus-stranded RNA, so it's a messenger RNA. But very much like the toga viruses, when that RNA gets in the cell, not all of it is translated, only the left half. So the left half is translated to make the polymerase, actually, very much like the toga viruses. And then the polymerase copies the plus strand and makes mRNAs, which then encode the structural proteins. And that, of course, ends up building new virus particles, which exit the cell. So the RNA is only here's the whole genome here in part B. And you see only the left-hand part is translated initially, and then the mRNAs have to be made to get access to the structural proteins on the right here. And those mRNAs, of course, are copied from negative strand RNAs. So this is a very unusual mechanism for doing this. Okay, so here we have the coronavirus genome at the top with some signals that are important for RNA synthesis. So the RNA polymerase, this gray oval here, binds to the 3' end and begins to copy the plus strand and makes a minus strand. All right. This RNA, the plus-stranded RNA, folds back at the very 5' end of the genome is a leader sequence. It's shown here in gray, and that leader is copied during the synthesis of every mRNA, so that every mRNA has a leader at its 5' end. And we think it's copied when it folds back here, and the polymerase switches from copying the minus strand, the plus strand, to copying the very 5' end. So you end up with mRNAs that have leaders, and then those are copied again to make plus strands because they're minus strands. And so you make mRNAs of different lengths. So the polymerase terminates at these termination signals, these TRSs, these blue signals here. And whenever it's terminating, then it transcribes the leader mRNA. And so this is a very unusual mechanism for mRNA synthesis. It involves the polymerase switching templates, right, from the 3' end to the 5' end. And so it's a kind of recombination. And this is what we think allows for high rates of recombination among coronaviruses and gives rise to coronaviruses with different biological properties, right? So that's very, very unusual. And we call this nested mRNAs, right, because they're overlapping. And that's really unusual. But also, only the first open reading frame of each mRNA is actually translated to a protein. So this long mRNA encodes a lot of proteins, but only the 2A protein is translated. Then the next one, only the HE, only the spike, only 4, etc. So only the first open reading frame is translated. So each of these mRNAs encode multiple proteins, but only one is actually translated. Now, negative strand viruses have a totally different approach because negative strands, of course, cannot be translated once they enter a cell. So they have to be copied first into a plus strand or a messenger RNA that can be translated to protein. And the cell cannot do that. The cell has no enzyme to copy RNA. And so these viruses like VSV and influenza virus, they must carry in the virus particle an RNA polymerase that will copy the negative strand genome. So we will talk about two kinds of viruses that have one RNA in the particle, we call unimolecular, and viruses that have multiple or segmented RNAs, influenza virus. And the common feature between these two kinds of viruses, there's a negative strand in the virus particle, and the virus particle brings in an RNA polymerase into the cell. The polymerase makes mRNAs from the negative strand for both the unimolecular and the segmented RNAs. And then the same polymerase also copies the negative strand to a full-length plus strand and copies it again to make more minus strands that are needed for making new virus particles. So let's take a look at this in some detail. Here we have VSV. Again, the genome is not mRNA. So it comes into the cell coated with proteins, including the RNA polymerase. The polymerase makes a series of subgenomic mRNAs, which are translated into the proteins and give rise to new virus particles. And among these proteins are the polymerase, the L protein, which copies the genome, of course. Now, these mRNAs are too short to be copied to make a genome, right? The genome is the full length, and the mRNAs are just parts of it. And so this brings up a rule, and that is when the viral genome is not mRNA, which it is not for minus strand RNA, there must be a switch from mRNA to genome RNA synthesis. So initially you make mRNAs and you make proteins, but at some point you need to make genomes, right, to put into virus particles. So you have to switch from mRNA synthesis to genome. And I want to tell you how that happens. So here's the architecture of VSV. It's a bullet-shaped virus. It's enveloped. The RNA is inside, wrapped in a helical nucleocapsid bound to protein, and there's a molecule of RNA polymerase in the particle. The genome is minus-stranded and encodes 5 mRNAs that encode proteins. And so when that RNA gets into a cell, that's shown here in the olive color, it's coated with nucleoprotein. The polymerase, which is attached to it, makes mRNAs. It makes all of these subgenomic mRNAs, which go on to make proteins. And early in infection, that's all that happens. mRNAs are made and proteins are made. But later in infection, the virus switches from making mRNA to making full-length minus strands to be packaged into new genomes. So how does this switch occur? It's governed by this circle here, this N protein or nucleoprotein. Early in infection, there's not much nucleoprotein around, but once you make the nucleoprotein mRNA, the levels of nucleoprotein start to accumulate. When they reach a certain level, they switch the synthesis from mRNAs to full-length plus strands, which are not messenger RNAs. They're just templates for the synthesis of minus strands. So that's the switch. It's governed by this RNA binding protein, the N protein. Here's a detailed view of what's going on. The polymerase is to illustrate how the different mRNAs are made. So here's the genome with the N and the P and the M genes, and there are more. They're not shown here. The polymerase is bound to the 3' end. It initiates synthesis at the 3' end, makes the N mRNA, and then it stops and restarts, and then it makes the P. So it makes individual mRNAs by this start-stop reaction, and it terminates at this intergenic region between genes. There's a signal for the polymerase to stop. So it stops, it's made an N mRNA, and then it starts again and makes a P mRNA. And then it stops, It starts again and makes an mRNA. And here's a close-up view of that intergenic region where the polymerase stops. So not only does it stop, but the polymerase adds poly A to the 3' end of the mRNA. So in this intergenic region, which has a stop signal for the polymerase, there's also a stretch of U's. And when the polymerase hits this, it starts to stutter. It keeps making A's and A's and A's until about 200 A's are added, and then finally the messenger RNA falls off, and it's been polyadenylated. It now has a 3' poly A sequence. And so that's how you get poly A tails added at this intergenic region. Now, influenza virus, similar idea, except the genome is segmented, right? It's in pieces. They're negative strand pieces. They have to get into the nucleus. They're copied into mRNAs, into nucleus, which go out and encode viral proteins in the cytoplasm. And again, the genome is not mRNA, so there has to be a switch from mRNA to genome RNA synthesis. And what is that switch? I'll show you. Here's the configuration of the genome. 8 negative strand RNA segments from which messenger RNAs are made that encode proteins. And so you see these messenger RNAs they're actually shorter than the genome. It's shown here. Here's the genome, the minus strand that comes into the cell. It is copied by the RNA polymerase to make a messenger RNA. The primer for mRNA synthesis is a piece of host cell mRNA, so that's stuck on the 5' end. And then the polyadenylation happens short of the 3' end of the RNA. So we're missing some sequence. So very much like VSV, the subgenomic mRNAs can't be used to make new minus strands. They're too short. At some point, we have to switch to full-length plus strand synthesis. And again, the nucleoprotein controls that. When levels of the nucleoprotein rise, then you start to make full-length plus strands rather than mRNAs, and you can copy those to make minus strands. Now, the primer for influenza virus mRNA synthesis is a piece of RNA that is snatched from host messenger RNAs. So here's the sequence of a host messenger RNA. There is an enzyme encoded by influenza virus that cleaves these mRNAs, and they can serve then as primers for the synthesis of mRNAs on the minus strand template. And the enzyme that carries this out, this cleavage, is an influenza virus enzyme. It is the target of an antiviral called baloxavir. It's a relatively new antiviral that inhibits the endonuclease that does this cleavage. And this antiviral is supposedly better than Tamiflu. The influenza virus polymerase has 4 different subunits here. You can see the 4 of them here. It's got a cap binding domain. It's got a catalytic domain. Here's the endonuclease domain that cleaves the host mRNAs and has binding sites for viral RNAs. So, for example, here's the minus strand genomic RNA. When it's going to be copied, it binds to the polymerase at the 5' end. At the same time, the polymerase is taking host mRNAs and cutting off their caps and using that as a primer for the influenza virus polymerase. And then the 3' end of the viral RNA will bind the polymerase, and this activates the polymerase to copy it. Now, when the mRNA is being made, it's made, of course, on the minus strand template, but the minus strand is locked onto the polymerase, and the 3' end is pulled through the active site. Here's the active site right here, and as it's pulled through, you synthesize the mRNA. So this is interesting because the enzyme doesn't move down the minus strand template. Rather, the template is pulled through the polymerase. And the consequence of that is that at some point, because the 5' end of the negative strand is locked onto the polymerase, you now can't pull anymore. And you're short of the 5' end. So that's why the mRNAs don't copy the whole genome. But there's also a sequence of U's at this part of the minus strand. And so what do you think happens? Yeah, the polymerase begins to stutter because it can't get any more template, and it's just cranking out A's. And yeah, that's how these mRNAs are polyadenylated. This is very cool mechanism. Hey, we have a quiz for you. Let's see how you do tonight. Frankie is not here, so somebody else can be the first. Are you ready? Are you ready? How are influenza virus and VSV RNA synthesis similar? The switch from mRNA to genome synthesis is controlled by an RNA binding protein. Polyadenylation occurs at a short stretch of U residues. Viral mRNAs are shorter than the minus genome RNA. All of the above. So how are the two similar? D. So Visto was first with D. Nevitz is D. Tom is D. Soul Symphony D. Vanity Nutrition D. Yeah, that was pretty easy, right? RNA does what to the genome? I got lost there. Okay. Viruses Luna says E. Hey, that's not funny. There's no E. And just a little bit more here. Double-stranded RNA viruses. They have a plus and a minus strand, right? Because they're double-stranded. But the plus strand cannot be translated by ribosomes. So these viruses have to bring in a polymerase to copy the minus strand and make messenger RNAs. And some of those are translated and some of those are replicated. And this is the configuration of the genome, the double-stranded RNAs. It's in segments, and each of them gives rise to an mRNA that encodes one or two proteins. Now, remember last time we talked about how this is a double capsid virus particle where there's an outer shell and an inner shell. And as this virus moves through the endocytic pathway, the outer shell is removed and it exposes these turrets. And then the mRNAs that are made inside of the particle can actually exit via the turrets. So here's how that happens. The virus is taken up by endocytosis. The outer membrane is stripped away. The particle is highly hydrophobic. It pokes out of the lysosome. And then we have this core in the cytoplasm. And the polymerase is active in the core and makes mRNAs, and they come out from these turrets. They give rise to viral proteins that can be used to make new virus particles. And this is a picture of a double-stranded RNA virus, rotavirus, synthesizing mRNAs. And you can see they're coming out from these turrets at the five-fold axes of symmetry. There's apparently a single molecule of RNA-dependent RNA polymerase underneath each turret at each five-fold axis of symmetry. And finally, last topic, these RNA viruses make a lot of mistakes. Well, all polymerases do. But they don't have proofreading activity. So you have a mistake for every 1,000 or 10,000 nucleotides polymerized. And so in a 10 kb genome, a mutation frequency of 1 in the 10,000 would give you about 1 mutation per genome. So if you make 10,000 virus particles in a single infected cell, which is not unusual, every base in theory in the genome should be mutated. Now the exception are the nidovirales. This is the order which contains the coronaviruses. They have an error-correcting protein called the XON, which is an exonuclease that corrects errors. It detects when there's a mismatch made by the RNA polymerase, and it takes it out so it can be fixed. And if you take the gene out of the genome, you make a virus that has a big increase in mutation rate. So it shows you that this error-correcting protein is really important. And this may allow the production of very long genomes. We found these nidoviral genomes up to 40 kb in length. That's huge for RNA, and maybe it's because of this error-correcting machinery. In poliovirus, there's a particular fidelity checkpoint in the actual structure of the polymerase, and this is determined by how the template, the primer, and the triphosphates interact at the active site. So you first have NTB binding in a way to the template, in a way that doesn't allow the ribose to interact with the asp acins. So here we have a UTP in the active site. So here's the ribose, and it's not interact with these asp acin residues here. But if the NTP is correctly base paired with the template, it undergoes a conformational change in the enzyme. It reorients the triphosphate and allows these interactions, and then the base is incorporated into the RNA. So this is how errors are managed in the polio RNA polymerase, although they still occur, and still you get a lot of diversity. And a single amino acid change in the poliovirus polymerase right here in this amino acid 64 causes it to make fewer errors. And we think it slows the conformational change that occurs when the right base is in there. It reduces the elongation rate and reduces the errors. And so this is in the fingers domain of the polymerase. It's away from the active site, which is here. But again, the change makes the enzyme more dependent on correct base pairing, which increases the fidelity, and this is probably conserved among RNA-dependent RNA polymerases. And these enzymes can also undergo RNA recombination, very much like the coronavirus, right? You have a polymerase copying one RNA. It can switch to another template so that you make a genome that is a composite of the original two RNAs. And of course, this happens if you have two different polioviruses infecting a cell. And this has certainly shaped the evolution of the RNA world. And it can be very high. 10 to 20% of all genomes in a polio-infected cells can be recombinants. Recombination is also regulated by the polymerase. There are 4 amino acids here near the active site, near the RNA exit channel, and they probably contribute to distinguishing between the homologous and non-homologous templates of the polymerase. And so that ends our little foray into RNA synthesis. I hope you get a better understanding of this unique reaction, right, which only happens in cells that are infected with RNA viruses. Okay, back to your questions. How are we doing with the likes up? All right, 106 out of 134 likes. Not bad. Yes, mini lecture time. You got it right in time. Right in time. So MedCram suggests the first 4 hours of sleep are most critical for your immune system. Okay. My immune system seems to be pretty good. A cat makes you sleep well. I don't have a cat. 2 shots of vodka. No, I can't do that. Ezra Klein or Rick Steves puts Polka Dot Pup to sleep. I don't have a sleep mask, no. But you can listen to music, huh? Or Sleepy Scientist podcasts. Oh my gosh. It might be helpful for some folks if you occasionally did pods about recording good audio and video. Huh. I didn't think people would be interested in that. Tona bought a December 22 ticket by doing a Google search for Immune 100. Well, thank you. We'll see you there. Mini lectures are interesting, like being a student again. Vanity says you can boost your body's melatonin with B6 vitamin C, AM sunlight exposure, and maximizing darkness at night. It's the higher melatonin, low cortisol that give the unwound feeling that promote rest. You know, I don't know that I have that unwound feeling. I really don't know. If you go to the Microbe TV website here, microbe.tv, let me show you. Come on, Microbe TV. I'm still waiting. There you go, right at the top, donate to Microbe TV, and below it here, buy tickets now. So if you click on that, you get taken to the Eventbrite thingamabobber, okay? Maybe some of you are not seeing this on the front page. It's possible, because we have funny things going on with updating. Could RNA polymerase be a target? Oh, I got the wrong thing on. Sorry. Could RNA polymerase be a target? Yeah, it is for antivirals, for sure. There are many inhibitors that hit the active site or outside the active site, like remdesivir, molnupiravir, right? Is there any legit reason for a polymerase to be in the cytoplasm? Well, for many RNA viruses, they don't need to be in the nucleus. They don't get anything from it. Except for influenza virus, there's nothing in the nucleus for them. So they stay in the cytoplasm. Yeah. So there is a legit reason for that to be. Powassan virus. Powassan. I'll talk about it next time. How's that? We'll do a Powassan little thingamabobber. Thank you for hitting the like, Patricia. Appreciate it. We got 113 out of 127. Should coronaviruses and togas be called positive when they're half plus and half minus? Well, it's actually all plus, because it's just that the plus at the end is not translated. But what's the most definitive thing is if there's no polymerase in the particle. That makes it a plus strand. There are some viruses that clearly have plus and minus in their genome, right? They have to have a polymerase in the particle to access that minus. So those are negative strand viruses, even though they have a plus, 50% plus, okay? Can the transcription switch from one template to a different template? When it hits the termination? It doesn't. There's no recombination in negative strand RNA viruses, so that what you're saying never happens, Visto. Magnesium glycinate doesn't do jack for me, but I take it because it's crucial for vitamin D. Okay. Maybe it doesn't do jack for me either, but if I imagine it, that's okay, right? Yes, you didn't tolerate the CPAP. The hell with that thing. Well, I never tried one. I don't want to use it. I mean, if you sleep on your side, you can avoid, right? So the thing is, it's nice to sleep on your back. You get good neck support, but then you're going to get sleep apnea. But if I sleep on my side, I can avoid the apnea. So I try and do that. I mean, the problem is I was a back sleeper most of my life, and to change is really hard, but I'm working on it. Yes, all of the stuff I showed you took years of research, and that's what's in jeopardy in this country nowadays. This is great, Polka.pub. Learn to appreciate being awake at 3 a.m. You get brilliant ideas. No, I don't get brilliant ideas. What I do is get frantic, that there's so much stuff I have to do. I'm forgetting something. When it's dark and I'm barely awake, it's the worst time, because then I worry about everything. As soon as I wake up and get some light, the world is good. But not in those dark 3 a.m., 4 a.m. I can't handle that. No. Can't handle that. Isn't this what Robert Malone worries about? mRNA effect on our genome? No, he doesn't worry about that. He worries about him being well known, so he just objects to all good science. How does the XON detect errors? Mismatched bases, right? If you got an A with a C or a G, that's mismatched, and the XO can do. How it actually does it is a good question. It's probably got some kind of a pocket that will fit. It will fit the proper pairing, and if it's not properly paired, it doesn't fit, and that triggers it to cut it out. Something like that. Is there a linkage to the Nita viralis proofreading functions and the high recombination rate? No, but there is a linkage between this discontinuous mRNA synthesis, which is basically a form of recombination, and recombination. Yeah. Dr. R, you forgot a step in regards to the 5' end. Just kidding. I'm blown away by that lecture. Going to continue with the online course. Yeah, the online has more details. Yeah. Are there plus sense RNA viruses where all proteins are generated directly without polymerase? Yes. So the picornaviruses and the flaviviruses, the whole genome is translated without a polymerase. But then the polymerase is made, right, so that it can copy the plus strand RNA and make more of them. See you in Bangkok. An Ayurvedic massage will help you sleep. There's no neutrophil ticket. Well, that was Kim, actually, their social media manager, and she just picked some names of immune cells. And the immune team was a little miffed that B cells are the most expensive, you know. Frank is here. The leading hypothesis is that eukaryotes have a lot more RNA virus than DNA virus because it's more convenient to not go in the nucleus. But, Frank, more RNA viruses are now being discovered in prokaryotes, so it's not clear that that hypothesis is correct anymore. Yeah, welcome. Nice to see you again. Razor said he knows 5% more than yesterday. Love learning, love my passion. It's infectious in a good way. Upon retirement, I'm going back to school. You're already back in school. Marge, hello. Yeah, we're almost done here. Amelia, Virginia, 13C. Frank says, I can't type anything, but I'm about to solve the structure of an RNA polymerase. There you go. There you go. Hour of the Wolf. Yeah, that's that 3 a.m. thing. The Anhatamama. Welcome. 27C in Western Massachusetts. From Mickey Spillane. Things always seem worse at time of night. That's exactly what I'm feeling. Everything seems worse. I feel nothing's going to work. Everything's tragedy. And then I get up and the light's on and I'm like, hey, everything's cool. Moving on. Yeah, I heard that CBD helps sleep also. I haven't tried that. All right, folks. That's it for another office hours. I'm full of energy. I could do it for another 2 hours. But I appreciate that you came to this and gave us your questions and listened. We had moderators today, Tom Steinberg. We had Les Fabi. We had Vanity Nutrition. And anybody else? Was Barb Mack here at the beginning? Let's go look. Don't remember. Nope. Thank you for moderating. Really appreciate your service. And thanks to all of you, about 140 of you that were here at one point. Really appreciate your being here and listening and asking questions and participating in the dissemination of scientific knowledge. We'll be back next week at the same time. Let's see what that's going to be. Today is the 15th. Next week is going to be the 22nd of October. October is almost finished, folks. Before you know it, going to be over with 2025. But in the next 2 days, I'll be in Blacksburg, Virginia, visiting Virginia Tech. All right, folks. Yeah, well, actually, once I went, Amy left because she had to eat and do some experiments. And I kept going for another hour. And I said I might do it now and then, but I never did. It's a long time to do it, and don't expect everyone to be here. But thanks for coming, everyone. We'll see you next week. And until then, please be curious. Bye-bye.